Huntington's disease is a fatal neurodegenerative disease, and it's caused by a kind of too much of a good thing situation involving the amino acid glutamine. This protein letter is really sticky. Basically, we'll talk more about this, but it can form these things called hydrogen bonds. These are the type of bonds that hold water molecules, like get molecules to stick one another, get the strands of DNA to complementary pair with one another. But they can also form with proteins, and glutamine is really good at forming these. What happens in Huntington's disease is this thing called a triple nucleotide repeat expansion. We'll talk more about this. But basically what happens is that in the gene for this Huntington protein, there's the CAG stretch. So CAG is basically there's the instructions for putting in glutamine. So when the cells are making this protein, they put in a glutamine when they see CAG. What happens in Huntington's disease is that they get a lot of these CAGs in a row, so the cells insert more and more glutamine. This can cause this the protein to have this long stretch of glutamine. It's like polyQ. Q's the or polyglutamine or polyQ. So Q is like the abbreviation for glutamine. So yeah, we use G for glycine, and so glue, Q, so that's how you remember it. But anyway, you get this long string of glutamine, and because glutamine is really sticky, now it can stick to itself, it can stick to other things, it can cause problems, it clumps up and forms these aggregates, it can trap things, it can slow down machinery. So it's not just as simple as it just being caused by the protein. There's also evidence that like the RNA itself can like fold up in these weird structures and like cause problems, all sorts of different things, and it's really complicated. But some of it can be explained by understanding this amino acid glutamine. So let's talk about glutamine. Let's talk about hydrogen bonding and repeat expansions, types of genetic mutations. So Huntington's disease is what we call autosomal dominant, and we'll get into what that means, as well as some of the work that I've done with HD in the past.
Okay, so this is glutamine. So it is an amino acid, a protein letter. It is the 18th that we're talking about in 20 days of amino acids. So if you've been following along, and even if you haven't, so basically amino acids are protein letters, and they link up through these peptide bonds, these amide bonds. So we call these amide bonds. Where you have this carbon, carbon, so carbon double bonded to an oxygen, bonded to a nitrogen. We call this an amide bond, or a peptide bond in this case, when they're between amino acids. But basically they link up into these long chains, and then they're going to fold up in a way that best accommodates the various side chains they have. So each of the amino acids, so each of these 20 amino acids that has a unique side chain or R group, and these have different properties, and those properties are going to influence how the protein folds. So basically the protein is going to fold up in a way that best accommodates each of these different side chains desires and preferences. When we talk about glutamine, it's going to be what we call hydrophilic. And so basically it really likes hanging out with water. So you often find it on the surface of proteins. And the reason why it really likes hanging out with water is because it can form these things called hydrogen bonds. So hydrogen bonds are just a special name that we give to a partial charge attraction when it happens to be between a hydrogen attached to an electronegative atom and an electronegative atom with a lone pair of electrons. So let's step back a second and talk and explain what the heck I'm talking about if you're unfamiliar with these concepts. So basically, each of these different atoms is made up of smaller parts. So each of these carbons, hydrogens, oxygens is made up of smaller parts, so subatomic particles. So there's positively charged protons, which hang out with these neutral neutrons in this central nucleus, and then they're surrounded by this cloud of negatively charged electrons. And so these are all whizzing around. And then when you form molecules, so when we have these, when you link the joined atoms together to form molecules, what happens is that you do this by sharing pairs of electrons. So they share a single pair to form a single bond, or two pairs to form a double bond. And these are what we call covalent bonds. So they're actually involving the atoms kind of like merging their clouds together. We can also have just like attractions instead of actual bonds. So when they're sharing electrons, they don't always share fairly. And so basically you can have partially positive and partially negative parts of molecules. So basically the protons, the positively charged stuff is stuck in place, but the electrons, the negatively charged stuff, can move around. And so if you have one of the atoms in that's supposed to be sharing this pair of electrons, if they share unfairly, so if they're really hogging the electrons, we call those atoms like electronegative. And a great example is oxygen, also nitrogen. So these electronegative atoms, basically they can pull the electrons, the shared electrons towards them, making them partly negative and the thing that they're attached to partially positive. And so what happens when we have a hydrogen bond is that we have an electronegative atom attached to a hydrogen. So this atom is going to pull electrons away from the hydrogen, making the hydrogen partly positive. Then you have the electronegative atom, and in the case of a hydrogen bond, it also has to have a lone pair of electrons. So basically, this is a pair of electrons that isn't being shared. And so you have this partial negative part, this partial positive part, and then you can get these like partial charge attractions. And these are stronger than most dipole-dipole attractions. There's a little bit of like covalent bond character in it. It's kind of complicated, but they're basically stronger than most partial charge attractions. And in order for them to be like the strongest, they need to be in a line where you have the hydrogen on, like, so you have your donor and the lone pair and the acceptor like in a straight line. And so this is part of the reason why water, when you like freeze it, so it expands, because it's trying to maximize all of its different hydrogen bonds. So you have the lone pairs that can act as acceptors, and then the donors would be the hydrogen bond or the hydrogen on the electronegative atom. And so in the case of water, you have two lone pairs that can act as acceptors, and you have two hydrogens that can act as donors. And so each water can form like 4 hydrogen bonds. And so you get these big networks of water. And when you freeze the water, it wants to maximize all those hydrogen bonds. And so you end up getting the water expanding, because when it's freezing, it's maximizing all of those angles. Whereas and this is going to make it like sturdier and stuff too. Like when you have like liquid water, the water molecules are like slipping past one another. They don't have the perfect hydrogen bonding to one another. So they can kind of like slip past one another and stuff. But you get this thing called the hydrophobic effect too, because of these hydrogen bonding and this like water stickiness. So basically the water is going to want to hang out with other waters because it can form all those hydrogen bonds to other waters. And so it's only going to let in other things that can like offer it some sort of charge or partial charge that it can form some sort of attraction to. And so we call these things hydrophilic, and then things that it doesn't, that offer it nothing, those things we call hydrophobic, and they're going to be clumped up together. And so we'll talk more about the consequences of this before, like we saw when we were talking about sickle cell disease. But that's not the only place that you can find hydrogen bonds. And a lot of things can hydrogen bond with water, and they can hydrogen bond with other things. So we see hydrogen bonding between the bases in DNA and RNA. This is what we call like base pairing, and it provides like the specificity. We also see it in the peptide backbone. So the peptide backbone of proteins, like where all the proteins are linked up. So it has these, you get these alternating donors, acceptors, donors, acceptors, donors, acceptors. And what's going to happen is that because you have this regularly arranged pattern of them in this peptide backbone, because all of these amino acids link together the same way, it's only this unique part that's going to change, then you can end up getting these characteristic secondary structural motifs, like beta strands, which when you link them, which can team up into beta pleated sheets, and alpha helices. So these are involving the backbone. But it's not just the backbone that can form hydrogen bonds. It's a hydrogen bond, remember, you just need to satisfy this where you have a donor. So where you have a hydrogen on an electronegative atom, so on an atom like an oxygen or a nitrogen, and then an acceptor, something electronegative with a lone pair of electrons, so something like an oxygen or a nitrogen. So anywhere you have one of these, you have the opportunity for having hydrogen bonding. And so when we're talking about it in the peptide backbone, we're talking about it in between those in this like amide group. So the carbonyl carbon, so the C double bonded to an O with the lone pair of electrons. And then that is going to act as our acceptor. And then our nitrogen with the hydrogen, this is going to act as our donor. If we look at glutamine, we see that it has an amide in its unique part in its side chain. When you link these up to form like protein chains, the amino group and the carboxyl group, they're going to kind of like join together in these peptide bonds. And so the amide is going to be like in the backbone. When you have glutamine, you also have an amide at the end. So we also saw this when we were talking about asparagine yesterday. So asparagine and glutamine. And with glutamine, the difference is we have a one longer linker. So glutamine is just like asparagine, but its linker is one longer, and this is going to give it more flexibility and kind of make it stickier because it can kind of like move around, stick to things more easily. But both of these, because they have this amide group, they can then form hydrogen bonds through their backbone, through their side chain, as well as through their backbone. And so they can form these hydrogen bonds to like other parts of the protein, to other parts of other proteins, to basically to anything that has the opportunity for making hydrogen bonds. And because they have both an acceptor and a donor in their side chain, they can form hydrogen bonds with either an acceptor or a donor. And so this is going to make it really sticky. And so now let's see an example of this at work in the case of Huntington's disease. So in Huntington's disease, basically what happens is we're going to get this long chain of glutamines in the protein, and this is going to cause it to misfold and clump up and cause problems. So it's a lot more complicated than just being that simple, but that's part of the toxicity is from this protein. Huntington's disease is a really awful disease, and it causes both cognitive and motor problems, neurological problems, kind of like the symptoms. It's sometimes described as like the symptoms of Parkinson's and ALS combined. And these patients gradually lose their cognition and motor functions, and it's a really horrible disease. And so there's a lot of research going on to try to find treatments and cures for this disease. And I highly encourage you to check out this website, HD Buzz, which I'll talk about later, to stay up to date on the research. There's some really great scientists running it. And if you are in the US, the Huntington's Disease Society of America, HDSA, does a really great job. They have a bunch of webinars and various ways to get involved and learn more about it. So let's talk about how this happens. And so basically Huntington's is what we call a dominant, autosomal dominant. So basically for every gene that you have, so the genes are like the recipes for making various proteins. And so with Huntington's disease, this is involving the gene for making this huntingtin protein, which has a ton of different like functions in the body, and it's this giant protein. It's like 3,000 amino acids. It's really big, and it can has all of these various functions, like helping regulate expression of things and all sorts of things. But yeah, I can never keep up with all the different things that it's purported to do. But the basic idea is that, like any of our genes, you have two copies of the recipe. Well, like any of our autosomal genes. So basically, the ones that are non-sex chromosome linked. And so you have, for each of these, you're going to get two copies. You're going to get one from your biological dad and one from your biological mom. If you have two of the same copies, we call it homozygous. So with these, basically for each copy, you can have a different version, and we call these versions alleles. So you get one allele from your dad, one allele from your mom, and these can be the same, in which case we call you homozygous, or they can be different, in which case you would be heterozygous. Sometimes if you can have diseases that are genetic diseases that are autosomal recessive or autosomal dominant. So basically, if a disease is autosomal recessive, that means that in order to like see symptoms, you need to have 2 copies of like the bad version, 2 copies of the mutated version. Whereas with autosomal dominant, you only need 1 copy to cause problems. So with autosomal recessive, you have to like inherit it from both of your parents, or there needs to be like a spontaneous mutation, so that you can, and then in order to like cause this, like so you didn't inherit it, but it happened like spontaneously to give you this mutation that wasn't passed on directly from your parents. And so if you have 1 good copy, the other can compensate, and so you'd be asymptomatic, but you have the chance of passing it down to a child who, if they inherit a second like bad copy or whatever from the other parents, then they would have the disease. So this was like an example we saw with sickle cell. We'll talk more about sickle cell again, because we talked about it before, and we'll talk about how it kind of is similar and different. And we can also have an autosomal dominant disease. In this case, you only need a single bad copy. And so basically 1 copy isn't good enough. 1 good copy isn't good enough. And so there could be a couple different reasons for this. Basically, one of them is this thing called haploid insufficiency. And so basically this is just like one of them can't make enough of whatever they need. It's not enough of the good thing to do whatever the protein is supposed to be doing. So basically, if the like bad copy is dysfunctional, and then you don't have enough protein to do whatever the functions are supposed to do. There's also dominant negative, or like gain of function. So basically the bad copy itself is toxic. So this can be for different reasons. Maybe it took on a new function kind of, and so it's doing stuff it shouldn't be doing. Another reason is it could be just like kind of hogging the stuff that the normal copy would normally be working on. So basically, like you can imagine that if you were to if a protein's function is to bind something and do something to that thing and then release it and then bind something else and bind another copy and do something. If you have a protein that is just binding it, but it can't do what it's supposed to do, it's kind of just going to like sequester that thing and cause problems. And so this is one way you can get a gain of function. And so there's some evidence for both of these types of effects going on with Huntington's disease, but there's a lot of thinking that there's like gain of function effects whereby the bad protein can kind of like sequester things, cause various problems that like so that gene itself is toxic, and it's not just the loss of the normal version. So amino acids are really awesome, but it's all about right place, right time, right number, right letter, all of the various things that are going to come together to make the optimal protein. And if you mess with that stuff, then you can end up getting diseases. And the way that you like mess with this stuff is by having mutations in the DNA instructions, so in the genes that have the instructions for making the protein. So there are different types of genetic mutations, and Huntington's disease is caused by like a repeat expansion. So basically, when we're talking about like inheriting 1 copy of each gene, so basically these genes have the genetic recipes for like making the proteins. In a process called transcription, there's like an RNA, a messenger RNA copy of the gene made. And then that gets like edited so that these regions called introns, so like the regulatory regions get cut out. The exons, the protein encoding instructions get glued together, and then these messenger RNAs are used by these protein making complexes called ribosomes. They travel along to make the protein, and they know what order of amino acids to link together because they're following the instructions in this messenger RNA. And these instructions are written basically in this series of codons. So a codon is like a A codon is like a 3-letter RNA word that's going to spell one protein letter. So basically, as the ribosome's going along, it sees whatever the codon is, and this tRNA molecule is going to bring with it that has the matching anticodon is going to come and bring it's going to be attached to the corresponding amino acid, link it up, do this a lot, you get this chain of this polypeptide chain, and then you release it. So the tRNA is going to know what letter to add based on the codon in the sequence. So if you have a mutation in the DNA, then you can end up with the mutation in the protein. And so technically the mutation in the protein is like an amino acid substitution. So some people will get like really pedantic about this. And like, if you call it a mutation, they'll be like, no, it's an amino acid substitution. The mutation is happening in the DNA. But we typically just refer to these amino acid changes in a protein as mutations. But not all mutations are going to actually cause problems. So basically, when we talk about this genetic code, it's degenerate. So basically, multiple codons can spell the same amino acid. So if we look at our table, we can see that in the case of glutamine, for example, CAA and CAG both spell glutamine. But when we're talking about Huntington's disease, we're talking about the CAG expansion, because it's going to be like basically the DNA copier is going to kind of mess up and accidentally like slip off, repeat, and put in the same CAG again type of thing. So you can get this expansion. And this is so it's not going to put in a CAA because it's basically kind of messing up and putting in what it already put in, because it slips off and it falls back on, and then it puts more of the same thing in there because it's following the template from the other strand. So it's not just going to put in a CAA. But if you put in a CAA, you get the same letter. You get a Q as well. But you might have it might not be as problematic because you also have the effects of like the RNA, the CAG structure of the RNA is different than the CAA structure of the RNA, even if the protein is spelled the same. So it can get kind of complicated. But the basic idea is that the genetic code is degenerate. So multiple letters can spell the same amino acid, but it's not ambiguous. So only one codon can only ever spell one amino acid. So basically, if you have a CAG, it's going to put in a glutamine. It's not going to put it in an arginine. So it knows what to add. But it's also non-overlapping. So basically, it has to be read in the correct open reading frame. So there are 3 options. So basically, depending on where you start, you'd get a different thing. And so the right one is signaled by a start codon. So when we talked about methionine, the amino acid methionine, if it's at the start of a protein, it serves as a start codon. And so that tells the ribosome to start there. What happens is you can get mutations that can cause problems or not cause problems. So you can have a silent mutation where basically you have no protein letter change. So you change the DNA, but not the protein. Although it could, as we talked about, it could still potentially be like not silent, but it's silent at the protein level. Missense, you would have one protein letter changed. Nonsense, you change this to a stop codon. So in addition to being a start codon, there are stop codons, and this is going to tell the ribosome that, okay, you've reached the end of the protein, you can cut it off now. So what happens is that if you get a mutation that turns what was a coding one, so what did spell an amino acid, into something that spells a stop codon, well, now you get sense would be if you were putting a letter in, but nonsense, so it's a stop, so it's no letter. So you have this nonsense mutation, so you would get like a truncated protein that would probably like get degraded or something. You can also have indels, so insertion or deletion of DNA or RNA letters. So this if you have a multiple of 3 that you add or remove, you don't get a frame shift, but you gain or lose letters. If this is happening in the region, so you gain or lose at least DNA letters. If this is happening in the coding region, so in a region where the protein is normally, like where there are protein instructions, this would cause you to have like different extra or fewer protein letters added. If this is happening in just like a regulatory region, then it might cause problems with regulation or that sort of thing, but it wouldn't like add protein letters or delete protein letters. So in the case of the Huntington protein, this with the repeat expansion we see is happening in a coding region. So it's actually happening in a region of the protein that actually spells for part of the protein. So it's not happening in the regulatory region. So it does add more amino acids. If you add them, it adds amino acids in frame. So basically it's adding multiples of 3. So it's just adding extra copies of the CAG. And it's not, if it were just adding like CA or something, what it would do is it would shift the reading frame. And so that would mess things up and it would put in different letters. And so this can cause like huge problems because you basically mess up everything after it. But with the case of the Huntington protein, you can imagine that when the DNA is getting copied in like replication, so each time that the DNA the cell wants to like replicate or whatever, or every time that's like transcribing, so it's making a messenger RNA copy, what's going to happen is that you are going to be copying this DNA. And if you have like a long strand of CAG, CAG, so even in the normal Huntington protein, there's these long CAG tracks. So people normally have like 6 to 35 of these, but in Huntington's disease, people can have like 40 or more. And so basically you can imagine that if the DNA copier was trying to copy CAG, and it slipped off and then it had to come back on, it might come back on and earlier and kind of like redo what it had already done. And in doing so, it would expand the CAG tract. If this happened in cells that get passed down, then the child could inherit a longer repeat than the parents. And so this is one way that you could have this disease occur even if somebody's parent didn't have the disease. And so there's actually like people with 36 to 39 CAG repeats, they may or may not get HD, and they're likely to get it later in their life. If people with 40 or more CAG repeats, they will get Huntington's disease if they live long enough. The age of onset is inversely related to repeat length. So patients with longer repeats tend to have earlier disease onset. I saw this really cool article that there was like something about the CAG, the uninterrupted CAG length itself. So if they had like a CAA, so it's still spelled Q, but it was interrupted the CAG, then that was protective, but I'm not sure what came of that research. But yeah, it's basically complicated. But anyway, so you can imagine that this could allow you get this thing like that the expansion length can actually like increase between generations. So that people might have an earlier onset, like a longer CAG length than than their parents actually had. There's also this thing called somatic mutation, or there's also this thing called somatic expansion, where basically, so in addition to this happening, like when the DNA is being copied, so basically like brain cells, they live for a really long time without like dividing and stuff. But you can still have this tract expanding in the cells over their lifetime because like if there is like a problem and they're trying to repair the DNA or stuff. So basically you can get somatic expansion where this tract is actually expanding during the lifetime. And this is thought to potentially lead to some of why the disease occurs like later in life, that if you're expanding this repeat over time, then you're building up to these longer lengths over time, making more toxic protein, that sort of thing. And so I was actually studying this as part of a summer research program. When I was in undergrad, I actually did a summer research with an HDSA, so the Huntington's Disease Society of America, I got a fellowship for them to do some research with Steve Finkbeiner at UCSF and the Gladstone Institutes. I was working with this great scientist, Dr. Gabriela Novak. And so my project was actually, I was looking at the somatic repeat expansion. So basically, I was seeing if the CAG tract was actually expanding in cells. And so to do this, I was using this, like, trying to use this cool like nanobiopsy technique, where basically we have these cells, and I was taking this tiny little needle and trying to suck out a tiny bit of the intracellular fluid, and then sequence the genetic information to see if the repeat size was expanding in these cells. And so I spent most of the summer just trying to get this technique to work and trying not to break too many needles. So I didn't get like strong results or anything, but I helped develop protocols for using this technique. And I learned a lot, and it was part of the big inspiration I had for doing a PhD program. And it also introduced me to the HD community, which is super, super great. I will forever be grateful to the HDSA, to Dr. Finkbeiner and Dr. Novak, as well as to everyone in the lab who really helped mentor me and even helped me design my experiments, help give advice for applying to grad school, various things. So I'm just so grateful. Mentorship is so important. And the people I met there were really life-changing, as were the people that I met through HDSA, so both the scientists and the families. So thank you to everybody. I highly encourage you to check out HD Buzz. You can follow them on like Twitter and stuff too. So it's run by a couple of really great scientists, Jeff Carroll and Ed Wild, and they do a really great job of explaining concepts in a way that is understandable, but they also like really know their stuff. They're really tight with the HD community, and it's a really great community of people who all care a lot about both the people and the science and making the world a better place. So highly recommend them. But I saw this cool article where basically these scientists have developed this, like a tracker that could actually see the clumps of HD in the brains of people with Huntington. So basically this like PET tracer. So what's really cool about this is that they could see it in like living mice, and that like, so basically in most cases in the past and stuff, they've looked at these clumps of protein in like autopsy brains and stuff, but if you could track them throughout the progression of the disease and stuff, it could help scientists really understand the disease better, as well as see if like in trials and stuff, if some treatment is working, because it can be really hard to tell if a treatment is working. So if you don't have some sort of like clear objective thing that you're measuring. And so now they're going to investigate this in people. And so it's exciting to see what's going to come of this. And so thank you to Dr. Rachel Harding for writing this. And do encourage everybody to check out HD Buzz. So how does this compare to another genetic disease that we've talked about? So before we talked about sickle cell disease, sickle cell anemia, and that is caused by a missense mutation. So where you basically have one protein letter changed. And so with Huntington's disease, we're talking about a repeat expansion. So we're talking about like an insertion, basically. With sickle cell disease, we are talking about a missense mutation. So we're having an amino acid swap. This swap is going to be from a glutamate. So from this hydrophilic residue. So a residue is basically when we link amino acids up then the remainder is like the residue. So when the amino acids link up, they're technically not amino acids anymore because they've used up their amino and their acid part to join together. And so then we can call it a residue. So basically, normally there's a glutamate here, which is hydrophilic. When you have this sickle cell mutation, it changes to a valine, which is hydrophobic. So it doesn't want to hang out. The water doesn't want to hang out with it. And this leads it to, so hydrophobic regions tend to clump together because the water is excluding them. So if we go from a glutamate to a valine, now what happens is we're going from a very hydrophilic to a very hydrophobic. Then if these region, it can find a hydrophobic pocket on another copy of the hemoglobin, especially in the oxygen 4 state. This causes it to kind of link up into these long aggregates. So, and cause problems. So with sickle cell disease, we're seeing a single letter swap from a hydrophobic to a hydrophobic, and it was causing these problems. With Huntington's disease, we're seeing an insertion mutation where we're seeing basically we have the right letter, just too many of them. And so we're putting too many of these glutamines. As we saw, these glutamines can form these hydrogen bonds, and this is going to make them really sticky. And so then they can stick to things and cause problems.